Good afternoon, and thank you very much for joining us today. I would like first to thank the organizing committee for the opportunity to present in such a great meeting. I'll be talking about how machine learning can contribute to prognostic and predictive models in MDS. Here's my disclosures. I own a stock at Amazon. So in this talk, what I'll be covering at the beginning is what do we mean by artificial intelligence, machine learning, and deep learning? And then I'm going to talk about why do we hear a lot today about AI and what are the advancements that led to this revolution in our time. And then finally talk about how do we use artificial intelligence in MDS research. So what do we mean by artificial intelligence? Artificial intelligence is making machine do things and think like a human without explicitly programming the machine. So if a human can drive a car, can I make the machine drive a car without me explicitly programming every step of that? Now, under the umbrella of artificial intelligence, there is machine learning. And machine learning is teaching algorithms and computers with data. There are two types of machine learning. There are supervised learning, where we know the answer. So let's say we're trying to build a model to predict response or no response to chemotherapy in cancer patients. We'll have one column that have the response or no response. We ask the algorithm to go and find patterns in the data that will lead to the answer. And then we hide the answer from the algorithm. And then we ask the algorithm, what is the answer? Is it a response or no response? And then we score the algorithm. The other part of machine learning is unsupervised learning, where we don't know the answer. In this situation, we ask the algorithms to find patterns in the data and collect the closest patterns to each other. A great example of that will be RNA-seq clustering. So clustering of large amount of data from RNA-seq, that's part of the unsupervised learning. And there is in between semi-supervised, which is part of between supervised and unsupervised. There's also deep learning, which is a subset of machine learning where the algorithm is using a neural network. These neural networks supposed to be mathematical modeling of the neurons in our brain. So in our brain, we have neurons that are connected to each other, and they fire up depending on what we do. And if we try to mathematically do that, that becomes a deep learning. The most common deep learning are convolutional neural network, which we use for computer vision, as you see on the left here. This convolutional neural network try to dissect the image into pixels and then kind of provide the answer. The other type of deep learning are recurrent neural network, and these are commonly used in videos and time series analysis. But one of the easiest way to think about machine learning is that you have a set of input. These inputs can be variable, images, text, videos. You have a set of output. This output is something we try to predict. So if we're trying to, there are two types of predictions. One could be classification. So we have an algorithm trying to detect dog or a cat. That's a binary classification. Or regression problems. These are continuous numbers. So trying to build an algorithm to predict the house prices in New York City. That will be a continuous number. And in between, you have a machine learning algorithm or you have a deep learning algorithm. So why today? There are several things that happened that led to the revolution of AI today. The first one is the computational power that derived from Moore's law. So Moore's in 1965 came and said that the number of transistors in each circuit will double every 2 years, which means that because of Moore's law today, we have a computer that's in the size of our hand, and that's the iPad. And this trend actually going to get even worse in terms of like we will have smaller and smaller circuits, more computational power, much cheaper computational power. The other thing what happened today is we have a huge amount of data that we start generating in healthcare. This type of data can be genomic data or clinical data. So for the first time in human history, we have such amount of data. And then the third thing, we have a lot of money pouring into the space. So these are the forces that drive what we see today in terms of the revolution of AI in healthcare. Now, it took us about $100 million and 10 years to sequence the first human genome, where today we can sequence the entire human genome for less than a few $100s. And why this is important, because what we see that the genomic data will be doubling every 2 years, which means that by 2025, or just a few years from now, this type of data will surpass YouTube, Facebook, and Twitter data combined. Now, this can be very problematic for the healthcare systems, because the question arises at that time: how do we store this data? How do we analyze it? How do we make sense of it? And this is where AI actually will be the only opportunity for us to mine this data. This is how medicine looks today. This is how I feel sometimes when I'm rounding on my leukemia patients in the hospital. But this is what medicine tomorrow, where for the first time in human history, we'll be able to collect clinical data, tons of omic data that we can run on our patients. We have sensor data. So let's say we have a Fitbit on our patients, we'll be able to collect sensor data, activity, and sleeping patterns and others, and then quality of life. And then the question, can we link sensor data to omic data, sensor data to clinical data? And can we blend all this data together to provide predictive model that can predict, for example, patients who will end up in ICU before they end up in ICU? Can we predict the side effect from chemotherapy before it happens? And this is why this time is very exciting to take full advantage of this technology. So how can we use AI in MDS research? One way we can actually optimize the use of the technology in healthcare is trying to take a disease like MDS, try to define the current paradigm, and then try to define problems in the current paradigm, and then target these problems, try to solve these problems using machine learning and AI. So as you know, today, the way we diagnose the disease, we take a bone marrow biopsy, we put on a slide, we identify dysplasia, and that's what the MDS. The next step is to stage the disease, and our staging systems are the International Prognostic Scoring System, or the revised version of IPSS, which IPSSR. The reason we do that, because we want to obviously divide the patient into higher risk disease and lower risk disease. And as you know and see, the treatment algorithms are different for those two patients. So what are the challenges today? Well, the diagnosis of MDS is very subjective. There have been multiple studies that show that about a third of those patients who get referred to a tertiary center in the United States with a diagnosis of MDS, they either don't have the diagnosis or vice versa. They told they don't have MDS, but actually they have MDS. Because diagnosing MDS on the pathologist's side requires very experienced hematopathologists. We have done a lot of work trying to study the prognosis of patients with MDS, and what we found that all our current model, what we predict in terms of the survival of the patient and what the actual survival of the patient is completely different. The other important point is that we might have like several patients in the same category. So let's say low risk IPSS, but these patients behave completely differently in terms of their survival, which means that there is a significant amount of heterogeneity in this patient population. Now, when we get prognosis wrong, we get treatment wrong. This is why prognosis becomes really important. And today we give chemotherapy. That's not just in MDS, but other myeloid malignancies and cancers, without even knowing which patient will respond to chemo. So there's a lot of uncertainty in our treatment. So how can we solve these problems? We'll start with diagnosis. So as I mentioned, if you take a pathology slide and show some dysplastic cells to multiple pathologists, what you found that these pathologists may interpret these dysplasia differently. So what you see on this slide, that this study asked multiple pathologists to think about, dysplastic changes in the myeloid cells, and you see the discordance between the blue and orange in some of those cells. There's a lot of subjectivity. Now, there have been huge advances in the computer vision in terms of using computers to see, for example, and especially in medicine. So in this study, what the investigators did, they took skin images, they ran them through a deep learning model called Inception version 3, and they asked a question, can the computer vision model predict whether this is a malignant or non-malignant melanoma? And the computer vision model was actually able to distinguish those images in somewhat accuracy similar to dermatologists. This is why today we live in the best time in human history, because we don't have to invent the wheels. what we can do, we can change the first layer of this model to now, instead of melanoma images, take normal MDS or AML cells, and then change the last layer to ask this model to predict whether the slides of the patients have MDS or AML. Now, the promise also of this technology is not just on the diagnostic part, but the question becomes, can I take an image, for example, and predict the molecular profile of the patient? So if we think that morphology is predictive of biology, can we look at the slide and predict which patients will have TP53 mutations? Can we extract features from the slide that can predict survival of the patients? And this work is currently ongoing. But based on that work, we ask a question, do we really need a bone marrow biopsy in the first place? So in this study, we took a large cohort of patients with MDS and other myeloid malignancy. These patients were diagnosed at the Cleveland Clinic and University of Pavia, which actually combined the training cohort. And this model, the final model was built, was validated in data from Munich Leukemia Laboratory. We run these data through a machine learning algorithms. We extract the most important features. In other words, we ask the algorithm, what are the important features that impacted the algorithm decision? We rank those important clinical features from the most important to the least important. And then we take these features and we build the final model. Now, what you see again on this patient cohort is that we have MDS patients from other myeloid malignancies that can actually look exactly like MDS and sometimes really hard to differentiate. So in this study, we use only CBC and next generation sequencing panel on peripheral blood. So the question was, if I use the CBC with differential and the next generation sequencing panel, can I distinguish MDS, for example, from ICUS, CCUS, and other myeloid malignancies? And what we were able to build a robust model that can distinguish these entities, as you can see here, with high AUC on the validation cohort. What was also really important that the model was validated externally on completely independent cohort to assure the reproducibility of this model in the clinical setting. The other important part about this model is that it's explainable. In other words, it will tell the investigator this is MDS, but also explain why the algorithm was thinking that this is MDS. The other thing can tell CMML, for example, again, explain why the algorithm chose CMML. So we use explainable AI to diagnose MDS without the need of bone marrow biopsy. What about prognosis? As you know, we have several prognostic models to certify patients in MDS. The most commonly used one are the IPSS and the IPSSR. The challenge in prognosis today that if we take, for example, 1,500 patients and try to plot them on this graph using the IPSSR scoring system, so we see the staging very low, low, intermediate, high, and very high. On the y-axis, we have the survival probability. So if you say, well, the median survival for patients with low risk is better than intermediate risk, that's true. This is the kind of like lines here. But if we take each patient as a dot, and the green one are the alive patients, the orange one are the dead patients, you see there are some patients in the low risk group actually behave worse than the high risk group. You see the intermediate risk group patients are all over the place. So the question we had at that time, our models, as you can see, there are differences between the predicted and the actual outcome of the patient. So can we build a model that can give us survival that's specific for a given patient? So to do that, we took demographic, clinical, and next generation sequencing data of 36 commonly mutated genes in myeloid malignancies. And we combined the data to train the model from a Cleveland Clinic and a Munich Leukemia Laboratory. We externally validated the model in data from a Moffitt Cancer Center. These patients got completely treated at a different setting, again, to assure the reproducibility in the model. But to further assure the reproducibility in the model in clinical practice, we validated in data from prospective clinical trial. We also validated during the disease course and prior to transplant for patients with MDS. We used a machine learning algorithm that had survival in it. And then we, as we mentioned before, extracted the most important clinical features, ranked them from the most important till the least important. After we finish building the model, we build this user interface that now we released for research only, where the investigators can actually put the clinical variables, hit calculate. And what you can see that the survival probability change based on changing the clinical variables. So in this scenario now, I'm making the patient very poor risk cytogenetics with TP53, and you see the survival probability change. What was also important that this model outperformed significantly our current model with IPSS-R and IPSS, even when we added mutations on the top of IPSS and IPSS-R for overall survival and leukemia-free survival. The other important thing is how much the model is getting the prognosis right. In other words, if we took the patients in the training cohort, we plot those patients on this graph. On the x-axis, you see the overall survival only for dead patients. So this graph is only representative for dead patients. And for the x-axis, you see their survival. So each dot represents a patient when they die. And then on the y-axis, you see the staging from IPSS-R. And you can see, for example, on very low risk patients, you see there are patients here that actually behave really, really bad, similar, like they didn't even live for a month. In the model, in red, these are the patients that were upstaged by our model correctly, where in blue, you see the patients who were downstaged by our model correctly. So you see that the model actually making right prognosis. But also what was important about this, that we had about 75 patients that were enrolled on the clinical trial from the North American SWOG clinical trial, which randomized patients to receive azacitidine versus aza plus berinostat versus aza plus lenalidomide. As you know, this trial was negative, and the trial was actually developed for higher risk patients. What was interesting that although this trial was designed for higher risk patients, about 68% of those patients, if you apply their data to our model, they are actually having lower risk disease by our model, not higher risk disease. What was also important that if you look at the median overall survival for patients with higher risk by our model and the IPSS, you see that the combination of azacitidine plus lenalidomide extended the survival, almost doubled compared to azacitidine. Now, this was not statistically significant, probably related to the very small number of those patients, but there is a potential that this trial would have been positive if we have a larger number of patients, where what you see, the median overall survival for lower risk patients per our model, not by the IPSS, the combination actually was harmful to those patients. So you wonder whether if we risk stratify those patients better on the trial would have been positive in this case. Finally, going to talk about predicting response or resistance to chemo. As you know, azacitidine and decitabine are FDA approved. The response rate is about 30 to 40%. It takes about 6 months. We have no idea which patient is going to respond. Try to build models that use clinical data, genomic data. We couldn't build a realistic model to use to predict response to aza and decitabine. So we decided we take the recommender system algorithm. So if you take your mentors and studied what movies they watched last week, the question, can we predict what movie they will watch tonight? So we switched mentors with patients, movie with genes, and the question was, can we predict response or resistance to chemotherapy? And based on using this association rules, we are able to identify genomic biomarkers, a combination of genes, that predicted resistance to chemotherapy in 93% accuracy, and those biomarkers present in about a third of those patients. Now, why this is important? Because we went back to the lab, we find leukemia cells that carry one of those three gene combination. We treated those cells with decitabine, and you can see these cells are primary resistant to decitabine compared to cells with just TIP2 mutated type. What we were doing in the lab, we were taking normal cells and knocking those genes one by one using CRISPR-Cas technology to try to study the mechanisms of resistance, how this resistance acquire by adding those genomic mutations together, and this work is still in progress. We then ask a question, we give chemo and the counts of the patient change. Can we monitor the change in the count to predict response to chemo? In other words, in order for Siri to understand the sentence, the sequence of this sentence matters. So can we use the same technology or algorithm to study the sequence of changes of blood counts and then try to predict response or resistance to chemotherapy? So to do that, we took a cohort of patients with MDS treated at the Cleveland Clinic and also Moffitt Cancer Center in the United States. And they received azacitidine or decitabine or combination. And we took their weekly CBC with differential for the first 12 weeks, the first 3 months. We also validated the model in data from University of Toronto to, again, make sure that the model is reproducible at a different clinical settings. We run these models through the algorithm, and then we extracted important features, as we mentioned before, and we built the final model. So the training cohort consisted of 288 patients, as I mentioned, that the combination of data from Cleveland Clinic and Moffitt Cancer Center. And these are the characteristics of the training cohort. And as you can see, it's a typical training cohort of MDS. And then we build the model, and what you see, the performance of the model on the validation cohort, you can see if we take data from CBC weekly for the first 3 months, we were able to actually predict whether the patient would respond at 6 months with area under the curve of 0.77. You see here, if you take the 90 days data, that goes up to 83%. This is a robust model. We never had a model that predict actually response to hypomethylating agents with similar performance. What also important about this model that also explain the output. So what you see on the left is a responder, and on the right is a non-responder. What was interesting to see that the algorithm was using eosinophil percentage, lymphocyte percentage, and RDW as part of other clinical variables to make a prediction. What you see here, for example, the changes in blue in the RDW made this non-responder. And this is important because we clinically never looked at these biomarkers before. And what you can see that the algorithm now highlighting important features that sometimes we overlook as a human. So in conclusion, AI will change our lives for sure. Already is doing that. If using your cell phone, you're using AI. As I hope I relate to you that AI will advance cancer research, certainly MDS research. There are still several limitations in the technology, especially when we apply it to healthcare, but I'm hopeful that these limitations will be resolved in the next few years. But also important to realize that this technology is not replacement of physicians. This is a collaboration between physicians and the machine. And when we wrote this review articles, and we were fortunate to be highlighted on the cover of the Lancet Hematology, what the artist actually demonstrated nicely, the collaboration between the machine and a human. So as a physician, I will be better physicians if I know machine learning and AI and embrace that. It's not a replacement for physicians. But the real conclusion is that the electric light did not come from the continuous improvement of candles. So if we really want to improve healthcare, we want to improve MDS and the outcome of patients with MDS, we have to think completely differently about what we're doing today. And I think we have to start embracing the technology and see how we can inject this technology in our care for our patients. So at the end, what I would like to thank all the organizers for the opportunity to present today. Thank collaborators around the globe. Without them, this work will never happen. It takes a village actually to do such work, from collaborators to Cleveland Clinic, to the United States, to collaborators around the world, my lab members that I don't have them anymore, and then our funders, the NIH, the Plastic Anemia and MDS Research Foundation, course, the biggest thanks for all our patients. So I would thank you a lot for being with us today, and we're happy to take any questions through the live sessions.